Welcome to our presentation for BS 2002. In this presentation, we'll be discussing a research paper that investigates the constitutive and regulated expression of telomerase reverse transcriptase in human lymphocytes. The aim of this experiment is to investigate whether transcriptional regulation of HTRT accounts for telomerase activity in human B and T lymphocytes. Telomerase is a ribonucleic protein enzyme essential for the replication of chromosome termini in most eukaryotes. It uses its own piece of RNA primer to elongate the chromosome ends by adding telomeres, which are G-rich nucleotide sequences, until DNA polymerase can move in and complete the lagging strand. This prolongs the replicative lifespan of cells. This is an essential step in the transcriptional process as it prevents the loss of genetic information. Telomerase is active in germline and cancer cells, and it's inactive in most normal and somatic cells. It consists of two essential components, HTR, human telomerase RNA, and HTRT, human telomerase reverse transcriptase, which is the catalytic subunit of the telomerase complex, with HTER transcription having little or no effect on telomerase activity. B and T lymphocytes, the major antigen-specific lineages of the immune system, require extensive cell divisions for their functions. There could be a potential correlation between telomerase and the cellular replicative lifespan of these cells. Recent analysis of HTRT have shown that expression is highly regulated during lymphocyte development, differentiation, and activation.
In this experiment, B and T cells were isolated from tonsils, thymus, and peripheral blood. Tonsils were obtained from patients with chronic tonsillitis, thymus is from children undergoing cardiac surgery, and peripheral blood from normal donors. Mononuclear cells of the B and T lymphocytes were isolated by immunomagnetic separation procedures, and flow cytometry was used to analyze the purity of the cells. This was achieved by carrying out 5 different experiments: activation of T lymphocytes in vitro, polymerase chain reaction based assay, Northern hybridization, polymerase chain reaction analysis of HTERT expression, and RNA in situ hybridization.
Activation of T lymphocytes in vitro consisted of activating CD4 T cells from peripheral blood. The PCR-based assay was carried out in 2 steps: telomerase synthesis and PCR amplification. Northern hybridization was carried out by extracting total RNA from purified cells for each sample and transferring them to nylon membranes, followed by pre-hybridization and hybridization. The levels of mRNA were analyzed. RNA in situ hybridization consisted of cutting sections of frozen tonsil tissues and hybridizing them using digoxygenin nucleic acid detection kit.
To analyze HTERT expression in B-cell subsets, cell lysate and mRNA were prepared from purified tonsil naive cells, germinal center, memory B cells, and peripheral blood-resting B cells, and were analyzed by TRAP-assay and Northern hybridization. Telomerase activity was detected at high levels in germline B cells and was potentially undetectable in naive cells, memory tonsil B cells, and peripheral blood-resting B cells. HTERT mRNA was detected in all subsets but higher in GCB cells compared to the rest. Despite the fact that high levels of telomerase activity found correlated with the elevated HTERT expression in these cells, it was also noticed that HTERT was also expressed in B cells of practically undetectable levels of telomerase activity. Thus, HTERT mRNA is present in all B cells isolated from tonsils and blood, with the highest levels in GCB cells. T cell subsets that were isolated from the thymus, tonsils, and peripheral blood were also analyzed. The findings show that telomerase activity was high in thymocytes, intermediate in tonsil T cells, and undetectable in blood T cells of normal adults, but showed low activity in T cells of children. HTERT mRNA was detected in all subsets of T cells regardless of telomerase activity. Thus, there was no strict correlation found between telomerase activity and HTERT in T cell subsets.
The results of this experiment provide insights into the regulated process of telomerase activity. First of all, the expression of HTRT is regulated during human lymphocyte lineage development, differentiation, and activation. However, it has been shown that HTERT is expressed in human lymphocytes independently of the presence, absence, or detectable levels of telomerase activity. Therefore, this result suggests the regulation of telomerase activity is cell-type specific and is controlled at multiple levels, including both transcriptional and post-transcriptional events.
Telomerase activity and its two essential components, HTER and HTERT, are regulated during both B-cell and T-cell differentiation and development and correlate with the levels of telomerase activity in these cells. However, the telomerase activity levels do not correlate with the levels of HTERT during T-cell lineage development.
Furthermore, two mechanisms that may contribute to telomerase activity in humans were studied: transcriptional regulation of HTERT and HTER, and post-transcriptional alternative splicing of HTERT. These two studies led to the conclusion there are various alternatives to the regulation of telomerase, as it can be controlled through gene expression at the transcriptional levels, which offers an economic and effective approach, and also provides alternatives for regulating its enzymatic activity through post-transcriptional regulation of HTERT transcripts. As there was no strict quantitative relationship between telomerase activity and HTERT or HTER transcript levels in lymphocyte populations, this suggests that transcription of HTERT and HTER is not sufficient for telomerase activity, indicating that other mechanisms contribute to the regulation of telomerase activity in human lymphocytes. I hope you enjoyed our presentation. Thank you.